Welcome, everyone. My name is Olivier Zablocki, and I'm a researcher in Matt Sullivan's lab at the Ohio State University. Thank you for joining us today for this week's Microbiome Informatics webinar. I know this is yet another Zoom meeting, but I sincerely hope that today's lesson will be of good use to you as we go through the weeds of analysis with Longread. So, as you've probably guessed, today is going to be about nanopore sequencing data analysis. And we will frame this technology and analysis of this kind of data in a viral metagenomic context, which has some specific problems that long-read sequencing can help solve. Quickly, in terms of the Q&A format for this session, for most of the talk, I will not be answering direct questions as we have a team handling the questions in the chat. But I can, of course, if there is critical things that do not come through, I'll be more than happy to help answering these here. Also, if you have questions for after the seminar, you can see here my email address. I'm also on Twitter, so please feel free to reach out to me if you have any lingering questions afterwards. Also, just one last thing, we have swapped our original meeting schedule. So if you thought that this was going to be advanced ecological statistics, this is not the case. Just want to put it out there just in case. And instead, Dr. Ahmed Zayed will be presenting after Ben Baldock at the end of this webinar series. But as of today, so we will be talking about hybrid short and long read metagenomics that we have developed as part of our Virion 2 pipeline that I will be talking about more shortly. So today I've got 4 objectives that I hope to go through with you today that I really want to teach you. So first, what is long read and specifically long read nanopore sequencing? There are several types of long read technologies. We will then go over its main features and what it can do and how it can be useful for your research. Then we will go into how this technology can solve problems that are particular to viral metagenomics. And finally, we will go into a little bit of a practice mode where we will go step by step into the processing of long reads as it comes out of the sequencer, all the way to assembly techniques to maximize short and long reads data. So what is long-read nanopore sequencing? So long-read sequencing is about 10 years old, and it's an umbrella term used to characterize sequencers that can generate, you guessed it, long reads. Long reads compared to the typical Illumina 250 base pairs, depending on the platform. And while there are an increasing number of available technologies, the 2 main players and technologies right now is Oxford Nanopore Technologies, sometimes referred as ONT, and PacBio and their smart sequencing tech. So these here, between these technologies, the way that they reach long reads are very different techniques. And therefore, because of that, they have very different informatic tools to process this kind of data. Each have their own challenges to reach high accuracy. And because a lot of it is actually software-based and not necessarily hardware, it is a fast evolving space. And so every couple of months, you have a tool that may be upgraded, sometimes to upgrade sequencing accuracy, or sometimes you have new tools that are taking advantage of the technology that nobody thought of before. And so we will focus obviously today on Nanopore, and full disclaimer, I don't work for Oxford Nanopore. I'm not a sales rep. and I invite you to go and read some great reviews out there where they describe the pros and cons of these technologies. They explain how they work in a lot of detail, and this could help you sort of focus on how this can help you. A lot of it is depending on your research questions, your budget, and the scale that you're looking for. And based on these factors, you can choose what's more appropriate for you. Now, let's get into the weeds of actually how nanopore sequencing operates, because it's important to understand how it works, to understand how the informatic tools that we will learn about later on, how they process this data. So essentially, nanopore at its core reads DNA in the form of electric signals. Perhaps you've seen this graphic before, where you have this nanopore embedded in a membrane, and you've got DNA on top and a single strand below, and you have a DNA read below afterwards. So very briefly, this works by first attaching a motor protein, here in green, a motor protein to your DNA of interest. And this motor protein has two major functions. It works first by guiding the DNA strand to the nanopore. Remember, on a flow cell, there is an array of nanopores, and you flood this flow cell with DNA molecules to which are attached these motor proteins. And they are guided towards the nanopores broadly through an ionic flow. And the motor proteins attached to the DNA will add as a sticker to the nanopore. It is also vital to attach this guiding enzyme, because it helps to control the rate at which the DNA unwinds through the nanopore. If there were no motor protein, actually, the DNA flow would be about 1 million bases per second as it goes through the nanopore. Therefore, we don't have time to capture the signal. And so the motor protein breaks this rate to about 400 to 450 bases per second. And this is continuously tweaked in the different versions of the nanopores that are being developed. And so one of the neat features is that this is actually from a bacterium. This nanopore is a re-engineered nature's design from a toxin, specifically an alpha hemolysin from Staphylococcus aureus. And the logo of the company is actually a top view of this nanopore enzyme, just as a tiny fun fact. I like to understand what I'm seeing, so I had to research what that logo meant in this context. And so the output of this is what we call a squiggle data, or what I like to personally call a squigglegram, where you read the DNA. As it passes through the nanopore, it generates a change in a current. And the DNA is read not base by base, but through 4 bases to 5 bases at a time. So 4 mers or 5 mers, for instance. And as the DNA goes through, this electric signal is recorded, and there is slight overlap, obviously, between each iteration of the DNA as it unwinds. This, in the end, generates a FAST5 file, which contains the electric disruption signature of each DNA molecule as it passed through the nanopore. Now, again, I'm not a sales rep, but I wanted to show you at least the range of sequencing devices that use this technology, because it caters to all budgets and scales. So you have from the lowest tier, called the flungal, where you have a tiny flow cell, which houses all the sequencing mechanism, including the nanopore array. And this has 126 nanopores and can generate about 1 gigabase of data. It is mostly used as a quality control device, or if you want to quickly check the sequence of a DNA in your sample, it is relatively cheap. and you can get fast turnaround real-time sequencing with that. Then you move up to the MinION, which has a higher capacity flow cell, about 2,000 of them, and can generate 10 to 20 gigabases. And when you combine multiple of these flow cells on a grid ion, you obviously quintuple the base output. And if you go even further, this is more like a sequencing center grade instrument where the flow cells have much higher throughput and can generate really a lot of sequencing data. So again, depending on your usage, your budget, and the scale of your experiments, you can sort of choose the instrument. And most of the labs, research labs, have this instrument, the MinION, probably the most famous ones, which strikes a good balance between output, price, in between the Flongle and the grid ion. So what can it actually do? And is it useful for you? So first, let's go with the major features of the tech. So you can achieve reads that are above 100,000 bases long. There are some records out there where labs really try to beat records, and 1 megabase is kind of floating around. You can also, and this is the only tech that can do this, sequence RNA directly, so you do not have a cDNA step. This can be particularly interesting when you want to sequence RNA viruses or transcripts that perhaps have epigenetic modifications. So these are essentially bases that have been modified and have some biological functions, for example, in terms of DNA protection with methylated bases. The technology allows you to recognize the electric signal of modified bases directly, either on DNA or RNA. It is indeed ultra portable for the MinION and the Flongle at least. So you've got these two pretty cool cases where they've sequenced microbial genomes in the middle of the Antarctic desert. And they've also tested the technology on board the International Space Station, where you can imagine it can be used for outbreak monitoring and stuff like that, or ensuring the purity of some of these instruments. And I personally dream one day it can be automated to go on the Mars rover. Why not? Lastly, there is also a new concept that they've released that I have not personally experienced, but they call it adaptive sampling. And this is essentially where you do not enrich your sample for a gene or sequence of interest, but rather you sequence everything. And in real time, the software will read the DNA and discard the sequence that is not of interest as it sequences in real time, so that you could perhaps do not have the need to prepare your sample too much beforehand. You can just tell the software, I'm looking for this particular sequence, and as it sequences, it will discard anything that's not that sequence. It's another way to do things, but I don't know much more about it. And so, is it useful? Does it live beyond the hype? Can you actually solve problems that we have as researchers in genomics and biology? So there's a very nice review that I highly recommend that is in the webinar resource pages called The Third Revolution in Sequencing Technology in Trends in Genetics. And it nicely summarizes some of the core issues that can arise when you want to do assembly with short reads. The first classic case is that short read assemblers struggle with repeats during graph construction using K-mer signatures. At the end of the process, the program is not sure where to place these repeats because there are repeats. So it struggles to determine the continuity in the graph path. If you have a long read, it will obviously cover a longer frame of the DNA molecule. And so even if there are several repeats along a long read, it could offer advantages. There's also the issue of GC bias, and that mostly arises when you have uneven coverage of certain DNA molecules during the library preparation. If you have a PCR step, many polymerases are known to have GC biases such that you will have these areas that are less covered in your short read datasets. Again, if you have a long read, this can be mitigated a bit better since a whole area rather than just a specific GC-rich area will be covered. And lastly, you have this issue of determining the variance of, let's call it here, a same species, or in this example, alleles from maternal and paternal inheritance. And what this does is, if you have a near identical strand of DNA and you have multiple copies of it that differ only in a single base or single nucleotide polymorphisms, sometimes the assembler will not know which to choose, and this may be determined by coverage, and it will select to resolve the graph path, it might select the one that has the highest coverage. Again, if you have a long read, this may be resolved since the entire length of the long read could cover these problematic regions. So I like to think of it that it is easier to piece back a puzzle if we have larger pieces. And this is essentially what long reads enable. However, you may have heard, it's full of errors, I can't rely on it, I'll wait until it gets better. It is true, it has issues in terms of sequencing accuracy, and currently the company claims that it's at about 97% accuracy by itself, only through software upgrades, so it has come a long way. From about 90% all the way to today 97%. And like I said, this is a fast evolving space. And while they are tweaking the actual hardware in terms of the quality of the nanopore that reads the signal, a lot of evolution is occurring on the software side, such that we may expect increments of accuracy over the coming years that hopefully will come close to the target of short read accuracy. But so far, we're not there yet. So I warned you that this would be framed in the context of viral metagenomics and how long read sequencing can help specific problems for this field. But before we get into that, I just want to give you a brief intro of the organisms that we're going to be sequencing. So a lot of major virus metagenomic analysis involve viruses of microbes in environmental samples, and these are most commonly called phages. And we now know that there are incredibly abundant, 10 to the 31 abundant, all over the globe. It's estimated that that is their numbers, and that they have critical roles to play in every ecosystem where they've been looked at. So you've got throughout the global oceans, they lyse, viruses lyse host cells about a third of all the host cells in oceans. This in turn has consequences in the recycling of nutrients throughout the global oceans and the trophic food web consequences thereof. In soils, for example, this is still a nascent field, but we are seeing a great specific soil virus diversity, as well as the presence of genes, accessory genes, most commonly called auxiliary metabolic genes, that encode functions that participate in the breakdown of plant-derived polysaccharides. So this could also have repercussions in the recycling of nutrients in soil. Increasingly, we also have a focus on the influences of phages in the human gut and other body sites. And lastly, in efforts to combat the rise of antimicrobial resistance, increasingly you have research or resurgence of research in phage-based therapy, in which you isolate phages that are specific to the bug that is antibiotic resistance. And it has showed great promise, and you already have a couple of success stories, such as this book, which are very encouraging for the future. So phages, but there are also the viruses in environmental samples, of course, but phages are the bulk of the organisms that you expect when you sample environmental samples. And when we look at the ecology of these virus communities in these samples, you usually look at it in 3 tiers. So on top here, you will have 3 samples, here named Community 1, Community 2, and Community 3. And each community has different populations, which you might call species. And you count, for example, the number of different species. Here, populations is the term used. And this is microdiversity. Within each population, you will have variants of each populations that may vary in terms of gene content or SNPs or host range, etc. And this is referred to as microdiversity, or simpler species variants. And so what we found is that sometimes short read based assembly can miss some of these population variants. And this has been confirmed using single cell genomics, where they essentially isolated host cells and sequenced each individual cell, and saw that for the same virus, you had these variants that co-occurred, and they compared their results to a regular metagenomics effort, and they were less present. They also confirmed this with long-read sequencing technologies, where they saw these additional variants for these populations. And within these variants, they also saw, apologies for that, they also saw the presence of niche-defining genomic islands, which we will get back to in a short while. And so you might ask, what is causing the short reads to miss this? Well, it's actually related to one of the 3 scenarios that I covered earlier, where you have the short read assemblers that struggle to choose which path to take if it is confronted to multiple options, whether these are repeats, and here microdiversity. So remember, we have microdiversity, which represents variance within a population. And so if you have enough microdiversity in a single spot, you can sometimes refer to them as hypervariable regions. And more often than not, these regions may coincide or overlap with a gene that is a defining characteristic of that population variant. And so what you would have here, for instance, is if you have 3 variants of the same population, and you have in your sample variant A, B, and C, if you have variant A that has the highest coverage in the sample, it will choose A, and so on with the others, if you have the highest coverage for variant C here. Apologies. And so the net effect of that is that you may have a chimeric construct of these variants, and so you might miss some interesting genomic features associated with that populations, such that it could be genes that are under active selection pressures, or this intrapopulation diversity can really reflect niche adaptation, for instance. And so obviously here we will go into more details for long reads, and the study I showed you before was authored by colleagues at the University of Exeter, with whom we co-developed a way to sequence viromes with long reads called virion. And so what they found is that using long reads, they compared the results to a short read data set. And what they found is that you have more complete genomes. The length of the genomic islands that could be recovered was significantly higher. And they could uncover some interesting biology in which they found that these genomic islands encoded interesting genes that could inform on perhaps some of the selection pressures that occur in the sample. And so recently I authored an update to that initial paper where we upgraded the analytics and the wet lab procedures in order to allow less DNA to be sequenced on the MinION sequencer. I invite you to scan this with your phone and it will take you to the bioRxiv paper. And actually the final accepted version of the paper should come out within a week or two. So please look forward to that. I will update the links in the webinar resources. And this is just to give you an overview of what we will learn today, which is we try to benchmark and see which tools work and don't work to make full advantage of short reads and long reads to assemble various metagenomes and see the improvements that we could gain, especially since, like I said, there are a lot of tools out there. They evolve quickly, and a lot of them are not specifically tailored for viruses. So we wanted to sort of assess a beginning of a SOP for looking at virus metagenomes. And so these are some of the a few examples of what you can expect when you add long reads, specifically from the second iteration of virion to a virome. So you have an increase in the recovery of genome sizes of the phages. Red is short reads only, green is virion 2 enhanced in the sense that we used both technologies. And here you have higher genome sizes, completeness rates are higher. You find microdiversity much higher than compared to short reads. And the number and size of hypervariable regions found within these genomes is also significantly higher. So let's get into the workflows and from electrons to genomes. Essentially, how do we go from translating an electric signal to hybrid assemblies? So first, we have to prepare the long reads. It's a bit different compared to Illumina, for instance, where you receive from the sequencing center or from the machine, you receive a FASTQ file, and then you proceed on to cleaning it. Here, it's a very manual approach. ONT provides very nicely coded automated software to do the base calling in real time. And base calling, essentially what it does is it translates the electric signals into a raw DNA sequence. But you can also have this almost in real time. As the electric signal is passed through the software, you can have the DNA sequence base call very quickly. Here, this will really be for high scale and specific to our study. So we'll really see the manual version of this, but automated way also occurs. Next, we also have the option to barcode our sample and to split these barcodes. So currently, what we've used and what is the most robust is to multiplex flow cells with up to 12 barcodes. ONT provides a set of 12 barcodes. And then we will move on to quality filtering after we've separated barcode data. After that, we will move on to assembly and correction. And so essentially here in this framework, we're going to work with the two technologies. With short reads, we're going to co-assemble them with long reads, with the hybrid function of the spades assembler. And in parallel, we're gonna assemble long reads by themselves and correct them, eventually guided by the short reads. Hopefully all of this will make sense to you at the end of this webinar. So let's go back to preparing our data and start with base calling. It all starts here. So like I said, each raw read that comes will be an electric signature, and this comes into the FAST5 format, which is essentially a modified file format used in engineering for a while that stores multiple layers of information into a single file called HDF5. And they basically converted to the convention for biologists, with FASTA, FASTQ, here FAST5. And this contains multiple layers of information pertaining to the electric signal for that particular DNA molecule. And what we need to do first is to translate this signal into nucleotide bases using a trained neural network model provided by the manufacturer. And they refine this model all the time. And this is in part what allows them to increase the accuracy over time. They've basically trained the model to recognize electric signatures compared to hundreds and hundreds of k-mer combinations that eventually the base calling software looks for overlap between these k-mer signatures to string together a DNA sequence. And at the end of this base calling procedure, you obtain a FASTQ file like you would be more familiar with. And so, again, you have multiple base caller versions and newer ones, older ones, depending on your workflow. We have used the Guppy, which is one of the latest base callers. And Guppy essentially houses multiple tools in one. And so here we will first use the base caller function of Guppy. It is relatively straightforward, but you need to give it key information for it to work optimally. So you begin by giving it the input file, which is here, the FAST5 path here for clarity. I've modified it this way. And then the output, this is the suffix function where you can tell it to put it in a directory with a specific name. So here I'll basically append the directory that will contain the base call data with the _ base call. Now, this is one of the most important parts. This is the configuration file. This essentially instructs the base caller on which molecule type you have sequenced, so either DNA or RNA. The POR version, so here this means that is revision 9.4.1 with a translocation rate of 450 bases per second. So this is the current Nanopore version and kit that was used to sequence. And flip-flop is the algorithm that you would like to be used in order to base call your data. And shortly after that, you further select options for the model within the flip-flop that you've selected. It's simply the name. And again, you have to really specify what you would like. Depending on the model that you give and the POR version, you may have different results that have been previously tweaked for certain types of data. So it is critical to know this information. If you have your results from an outside source, they need to give you this information if they don't base call it for you. You also have -v, which is verbose, just showing the log. It always helps to see what it did. The -r is for recursive, so you will get all your reads by waves of 4,000s. So you will have 4,000 files in each folder. And so you're just telling the software to go through all your subdirectories and to base call anything in this directory. -X is an option to use a GPU setup, hardware setup, instead of a CPU. And what this does is, if you have the hardware on a cluster or on your gaming PC, you can dramatically accelerate the speed at which the base calling is performed, since it uses CUDA cores, which are much better at working in parallel jobs. And lastly, the very last is --fast5out. And what that does is it appends the base calling information to the original fast5 file input. And this is sometimes useful for workflows related to epigenetic modifications, where it tries to look at variance in signals compared to a modified versus a non-modified base. And so it is used to recalibrate a bit that signal compared to a reference sequence. But we will not touch upon that today. I just wanted to show you that it is an option to create these augmented fast5 files as well. So now that we have a FASTQ file, it's still all the sequences output from the flow cell, but you may have a couple of samples in there. So you need to split the base call files. So like I said earlier, currently it is reliably doable to do 12 barcoded samples in one MinION run. To detect these barcodes, there are several tools, some provided by ONT, some defunct tools on GitHub, and probably new ones that will come up. One of the challenges to detect barcodes and bin barcoded data is to actually figure out if it's correct when you identify the barcode, because this barcode may have errors. Remember, this data hasn't been corrected yet. And so if you tell the barcoder that, you know, it needs to be 100% exactly the same as the theoretical sequence of the barcode, it's not going to work. There's still mistakes in those reads. And if you happen to have a mistake in the barcode region of the molecule, then you might have some problems. So this is a problem that has largely been solved. And so in the beginning, it was kind of an issue. But this is why there were also several tools that try to do this the most accurately possible. So I want to show you today two that I've used personally. So you have the one from ONT, the official one, called the Guppy barcoder function. And essentially here you have number of threads, the input, you know, master FASTQ file, the suffix appended to the output folder, recursive once again to look in all the subfolders, and you have to tell Guppy which barcode kits sequences it is supposed to expect, and will work based on these to demultiplex your sample. Another alternative that is now not supported anymore, but that I still use and many others use, is PortChop. And essentially this was one of the first ones before even the official Guppy barcoder came out. PortChop was the gold standard, so to speak, because it had these very good options where it required 2 barcodes for each read. So 5' end and 3' end of the DNA sequence needed to have these barcodes, and it looked at mishappenings in some of the physics of the library prep and the sequencing, where sometimes you would have 2 reads being read in a row, such that you would have a barcode in the middle of a read when you see it after it's been base called. So when it sees that, it will discard it too. And that was a neat function that now the barcoder also does by default, but you can tweak what you want. But I still use PowerShell out of habit, and it's in all my scripts, so you know how it is. But it works very well. Again, here you've got the input, the barcode directory, where it will output all of these folders, barcode by barcode, as well as all the ones that either of the programs couldn't decide. Remember, there's still errors there, but in my experience, it's very small. It's usually less than 1% of the reads go actually into the unclassified bin. You have a barcoding summary as well, which is just the output of the verbose option. So now you have your barcode bins, and you know which one is which in your data. Now we're not done yet. All of these bins still have errors, and this will be the equivalent step of Trimomatic and these Illumina related tools. So we need to filter these reads based on the quality score and typically their length. And so we also will trim the ends. We'll see why just now. And there are, again, many tools to do that. But by force of habit, I will show you Nanofill that has worked well for me. It's relatively easy to work with, and you can get started very quickly. So here, for instance, what you do is you have to concatenate all the reads into a FASTQ file first. So what I mean by that is, like Sharif taught us the other week, it is one of those commands in a Unix system where you can basically tell with a wildcard here, the star, to take all the FASTQ files. Remember, every read is an individual FASTQ file. So what you want to do for this workflow is to take all the individual FASTQ files and put all the reads into a single file. So this basically instructs concatenate every file with the fast.fastq extension and create a new file, which I've named originally all reads.fastq. Then we use the single file we compress the single file because not only does it help for your hard drive footprint, but also Nanofill, I believe, is coded to only take compressed data. I think the author did that by design to force people to systematically compress their data. And so what we first do is, I've broken down this process, but usually you can, pipe everything into one. But you would basically gunzip the file that we just compressed, and we pipe this to Nanofill, which we then instruct to discard every read below a quality of 9. We instruct also Nanofill to remove all reads that are 1 KB or lower. This is to ensure that we don't really keep noise. These pieces of DNA that may have broken up, that are not really supposed to be there. After all, this is a long read run. So most often you have reads from a couple of base pairs to maybe a couple of hundreds that is really just noise and garbage that you should discard. There's also the option here where you trim the first 50 bases of each read. So here they call it a head crop. And this is to ensure that the barcode has been removed. When you do the barcode splitting into bins, the software should normally remove the barcode sequence after it has done its job to identify and bin. But again, there might be some errors. So it may still remain a few bases from the original barcode that's not, your sample. So just to be on the safe side, a rule of thumb has been you trim the first 50. But actually this one is outdated here, the command. So you could also tail crop 50 base pairs. So this is basically removing the 50 bases at the other end of the read. And then after that, you would essentially here zip it back, compress it back, and like Sharif said in his nomenclature of naming all your files that you can follow, basically rename here cleaned all reads.fascia.jz. And so this is essentially a representation, just to give you an idea of the amount of data that we just filtered out. Here, this is a density plot where every purple dot is a read with the read length on the x-axis and the mean Q-score for every read. And so what you basically want is everything above 1000 of above 9. And this square that just departed represent about 70 to 90% of the usable data. So the filtering, you don't lose too much data in this filtering process. And again, as the tools recognize and get better, there is less data. Better data is generated in the first place, such that you don't have as much trash to remove downstream. All right, moving on to assembly and correction. Now that we have our clean data, let's try to piece back the puzzle, right? So we'll focus on two parallel pipelines that are intertwined in that they use each other's advantages to sort of remediate the disadvantage of each. So we will first start with long reads and the assembly and error correction pipeline, and finish with short reads. So for assembly, you have spades for short reads, but in hybrid mode that will essentially use long reads to scaffold short reads. We'll get into that later. But for now, we will look at minimap2 and miniasm, which represent the assembly step of the long read assembly. And then we will get back into the steps that remove as many errors as it can from these long read assemblies. Before we get into that, I just want to give you a visual overview of what these tools are actually going to do. If you have these long reads here, you will first have an assembly method called overlap layout consensus. This is not a new assembly method. It was actually, I believe, the assembly method similar to Velvet for 454 reads. What this actually does is it looks at overlaps between reads. It's the old school method before K-mirror graph methods appeared. Is the old school method to assemble long reads. And so it will just look at overlaps between reads and deduce a consensus sequence out of that. And in here, the red bars represent errors that are present along this OLC-based contig. And then once this is assembled, we're going to try to remove these errors. And we'll use two tools that are independent of short reads. The first one is called Racon, and this is followed by another tool that's called Medaka. Now, Racon basically uses all versus all read mapping information in order to try to weed out errors that occur along repeated rounds of self-mappings. So it's a bit, let me try that again. What Racon will do is it will essentially take the information from self-mappings of all the reads and look at the consensus sequence between all of these mappings. And from that, if you have an odd base that's not part of that coverage consensus, it will identify it as an error and take it out. All right. And so on the right-hand side is, and you can do this iteratively multiple times until you reach sort of a plateau where you've weeded out all the errors you could based on self-mappings and coverage. And here in the paper, in Viromics 2 paper, we show that after 3 rounds, it is enough to significantly reduce the number of errors. So this is the number of errors per 100,000 bases. You can see here that it starts at 1,500 errors per 100,000. That's at 0 rounds. So this is the number of errors that you have in the data before correction. You apply once RACON, there are more errors that accumulate because it's quite early and it's the first iteration. After the 3rd one, that mapping information is refined and it drastically goes down to 500. And what I tried is to see how far off you can go, and you basically have no significant differences in error correction after 3 rounds of RACON. And this is broken down in the 2 major types of mistakes that Nanopore does, which are mismatches, so SNP errors, and indels, so insertion deletions, which are very common. So these are insertional deletions of 4 bases at a time, 5 bases at a time, which happens when there's some sort of slip during the DNA translocation through the nanopore. This also has quite big impact on the ORFs that you call later on. So if you have too many mistakes on a config, it may call upon it. This may translate into a stop codon prematurely, where it's not supposed to be a stop codon in the middle of your protein code. And so what we saw is that uncorrected, the median protein size in amino acids is about 70 amino acids. Whereas here, this dotted line represents the median for short read based protein sizes. So you can see that there is indeed, these errors incur an impact in the sizes of the proteins that you want to work with, which has, as you can imagine, have a lot of consequences for downstream analysis. And so here again, you can see that you have significant differences between the 2, and afterwards it starts to plateau. All right. Now, let's continue. You have then, this is what I just explained with RACON, self-mappings. I will get into Medaka in just a little while. I don't want to overwhelm you too much. But we get now into the phase where we use the short reads to guide the correction of the long read assembly. So what you would do is you have your assembled long reads, and you do a mapping exercise where you map your short reads to your long read contigs. Then you take this BAM file, which contains these mapping coordinates, and a tool called Pylon will identify, assuming the short read consensus is correct, it will identify wherever region is mapped by the short reads. It will assume that the short read sequence is actually correct and polish the region of the long read based on the information it has from the short read. And so this is not perfect, but it will get you to about 98, 99% accuracy. And briefly, the hybrid assembly, although we'll get back to that a bit later, but essentially it is within spades, and what it does is it does not perform the assembly with both read types together. it performs the spades assembly as usual, graph-based with short reads, and then in attempts to resolve gaps using long reads to scaffold the contigs into final, products. So it is not used during the actual assembly process, but rather to also polish whatever gap there may be. So let's go into running the multi-step of long-read assembly. And so this is done assembly and polishing with Minimap2, Miniasm, and Racon. So here we have Minipolish, which is basically a Python wrapper that will iteratively apply Racon multiple times. It is a neat little tool on GitHub, which you can find in the webinar resources that has saved me a lot of time. But we'll get there. So again, like before, I've put the code in this manner to simplify the command. So you've got the output directory, the reads. This would be your clean long reads, .fastq, number of threads, and the sample name that you want to give. So Minimap2 is a mapper. It's like BWA, it's very fast, and it accounts for the mistakes that are present within long reads. And the first step of this all versus all mapping is done by Minimap2. This here is the number of threads, and this setting here tells Minimap2 to account for errors specific to Nanopore, which is this ONT setting here, the -x. And then this is both inputs it expects. So this is, let's map all these reads against all these reads. And here, of course, it's the same data. This will result in an output file with a PAF extension, which you don't have to worry too much about. But this is what this step does. It creates a sort of BAM file, so to speak, with all the mapping information that will serve as a guide for Miniasm, which is the assembler itself. And what this basically does is it takes the reads and the guiding information from the PAF file, and it creates a GFA file. So this is a very raw and basic assembly step, and the GFA file is called a graphical fragment assembly file. And what this does is basically it's a tab delimited text format that describes a set of sequences as well as their overlap. And so this will be fed, this GFA file is fed to Minipolish in order to apply Racon iteratively. Here, for example, you have Minipolish, you give it the number of threads, the number of rounds you want Racon to be run over and over and over on this same input GFA assembly file. And here, again, some sort of guiding nomenclature, output directory with the sample, and you append Minipolish, the number of rounds that you did for this file to be generated, and the output is here still a GFA file, which is not very useful to you. You want a FASTA file, preferably, and so you can use a rather simple awk command. Awk is a scripting language that is present within most Unix distributions. You also have a biology-centered version, BioAwk, that exists, but I don't think is installed natively. But anyhow, it basically removes some lines of that tab delimited file in order to transform the GFA file into a .fa file, which gives you the set of Racon polished contigs in FASTA format. And so now you have assembled your long reads and did Racon correction, and you have still 2 correction tools to go. So let's get into Medaka and Pylon. Medaka is a bit, I don't fully understand it myself, how it does it, but it's a neural network based approach where they've trained models to recognize long read signatures in order to maximize overlaps in order to correct. It's probably the worst explanation ever, but it does work a little bit, not great, but it does add. And I invite you to read about it elsewhere. But here, again, you have output directory, the path to the reads, the number of threads, and the sample name. You activate your environment or whichever way you like to use it. And here, it's rather simple. You have, this is how you use the command. So you will have your input reads, your directory for the output. I'm sorry, this is the output name of the directory. And very importantly is the model selection. So I talked about that Medaka is neural network trained for specific types of electric signals, so recognition. And so it is critical that you give the correct model that matches the way that you generated your data. Otherwise, it's an entirely different model that's been trained and it's useless. So here, for the model, it's called R941_min_high_G344. So when you run Medaka consensus and you do --help, it will list all the available model files that come with the program, and you can select which one matches how you generated your data. Again, this is extremely useful information to have. As soon as you receive your data, you need to have the information on how this was generated. And so here you can see that this is the poor version. So this is the R94.1. The device here, min, is the min-IN. The caller variant is the high accuracy. And the version of the caller that was used, here, Guppy, is version 3.4.4, for instance. And so you select that model. And lastly, you run number of threads, and it will attempt to correct solely based on, I suppose it tries to reapply or check the model predictions on your cold data. In my experience, it adds maybe 1% inaccuracy, which is non-negligible. Next, we're going to go into Pylon, and this is where we slot in the short reads. So we first have to do some data preparation for Pylon to do its job. So we have first, we have the mapping to do. Remember in the graphic before, we have to do the short read mapping to our long read contigs. So for this, we use a Burris-Wheeler algorithm, or BWA. You would module load that. You also module load SAM tools, which is a very popular tool to work with SAM files and BAM files and extract alignments and so on and so forth. Here, just examples, of course, you do what you wish for this to be the clearest to you in your code. But this is basically the names of the data sets for ONT, so the long reads and Illumina ID here. You just want to make sure that you have the correct corresponding short read data for your long read data if you have these data sets. And again, here, for sake of clarity, you have the long read path, the path of the forward short reads, the path for the reversed short reads, and the working directory. And so if you work with BWA before, you will know you need to index the target. So you would index the long read here. Onto the mapping itself, we use here BWA mem with 28 cores. and this is the input. So you have the long reads here that will be mapped against the forward and reverse short reads. This is then piped into SAMTools view to transform the resulting SAM file into a BAM file. And this line here, this code, actually removes all the reads that were not mapped, so that you reduce the footprint and the size of the BAM file. I've done this mistake myself. And when you don't use this very intelligently, you have a BAM file that's 20 gigs in size, and you just can't handle it further downstream. Anywho, so it's a BAM file. You filtered your BAM file. You sorted by order of alignments, and you output it here. I've named it .sorted.bam. And then you would index the file in preparation. This basically speeds up and indexes the file for downstream quicker access. And then we get into, sorry, I should have done that. And then we get into the actual correction from Pylon. So now we have all the files that Pylon needs to do its job. So the Pylon executable that I have, Pylon is compiled in Java and is basically just a one-in-all package. Here you can set the memory, its memory requirements. So for example, here you would modify this, and here I have 128 gigabytes. And then -jar, Pylon version .jar. And so here it's quite straightforward. The genome it expects, it's called a genome like that because Pylon was originally developed to one of the uses. It was developed for the Human Genome Project in order to resolve variants, I believe, during that sequencing effort. So it just basically expects a genome, but it will accept pieces. So it can accept thousands of different contigs. And so this would obviously be the long reads. You ask it to fix all. It can fix multiple types of errors, indels, SNPs, and so on. So you ask it to fix all the type of errors it is learned to recognize. This is the name of this is a log file, I believe. This is the naming of the output. I basically put pylon.correction abbreviated, and you tell it where the BAM file is and where to output the results. And that is pretty much it. And if Pylon is quite well coded, because if you forgot a step in the data prep, for instance, it basically, to a certain extent, detects the error in your input data. So for instance, when I first started to use it, I didn't know you were supposed to SAMTools index your sorted BAM file. So it actually detected, it looks like your sorted BAM file is not indexed. Try that. And that was it. So for that, it is quite useful, and the documentation is quite extensive. And so one of the logs I wanted to show you here for a single config. This is an example of what it shows that it did. So for a specific config here that was 22,667 bases long, present mapped against the sorted.bam frags is what Pylon calls a BAM file, essentially. And it shows you that it has a certain coverage with 2,500 reads, average coverage of 6, and could confirm that this amount of bases was covered by the reads. And it could correct 45 single nucleotide polymorphisms, detected 2 ambiguous bases, probably it was ends. And here is the insertions and deletions. So these are the indel errors. And so this is what it shows you. Sometimes it's not that clean and it just can't find any resolution because of not sufficient coverage, for instance. And when you run Pylon, you can tweak its behavior. For example, you can instruct it to, ignore a correction if it does not have a certain minimum coverage threshold value, for instance. And so we're almost at the end of this fascinating assembly journey, where we go to the parallel line of assembly with the short reads and hybrid spades. So we dealt with the OLC, so overlap layout consensus path, with long reads. So now we're going to use the long reads to augment the short read assembly. So one important aspect of this is that you do not need to correct the long reads if you're going to do hybrid assembly. And this is because, like I said earlier, hybrid spades will only use these long reads, which can be PacBio or Oxford Nanopore. It will use them for gap closure and repeat resolution, such that it should not be a huge problem. I mean, it can be for certain applications that require ultra high accuracy. But if you have a 2 megabase genomes that needs gap closure, it won't be a big issue, even if there are some indels and mismatches present. So it's also fairly straightforward to run spades. So you would run spades. Again, the path to the short reads, forward, reverse, input path to the long reads and the output directory. And what you do is you run spades.py and you add the Nanopore option, which also could be the -- PacBio. And spades has been tweaked to take into consideration the long read data. And I also invite you to go and read up on the hybrid aspect of spades. They have a very nice explanation and tutorial on their GitHub. And but essentially, you tell it use this set of Nanopore data. It will then, you can tweak the way you want spades to assemble your data. Here, it's forward, reverse, very simple. In this context for viral metagenomics, we use the -- meta option, number of threads. And I believe this is the amount of memory, RAM that you want to give spades, and the output directory of spades. And there you have it. You have hopefully an augmented hybrid assembly. Now you have corrected assemblies. Now what? Well, this is where I leave you in the good hands of Dr. Ben Bolduc, where in a couple of weeks, he will teach you all about the virus analysis in metagenomes and cyber infrastructures where you can run your analysis in the cloud, as well as all the wonderful virus-centric tools that we have from iVirus. And so I will not cover that, but hopefully I've enabled you to prepare the assemblies in order to start looking for viruses in your data. Next week, we'll have Dylan Croonen, who will go over the basics of how to process your short reads, all the way to obtaining metagenome assembled genomes or MAGs. So please look forward to that. And with that, that is all for today. I sincerely hope this will be useful to you. Thank you so much for attending and being with us. Feel free to ask questions in the Q&A. I will stay here if I can manage to find my way around this Zoom window. There's also my email, my Twitter, and I want to acknowledge the sponsors, funders, and my wonderful colleagues whom have enabled this work. And again, thank you so much. And that is all. Thank you. Kate, is there, how can we proceed from here?
Yeah, there's some questions in the Q&A. If you stop sharing your screen, you might be able to see them a little better. If not, I can.
All right, stop share. All right.
Someone's asking for your polishing programs. Did you look at nano polish as well?
Yes, this was one of the options, one of the earliest options. I have not tried it personally. One of the ways that I've looked into nano polish was for epigenetics, and I believe since then, as other tools came in and out, nano polish I think has been phased out a little bit, but I think it is quite useful for epigenetics. But to be honest, I haven't looked at the latest updates for this tool, but it's definitely a nicely documented tool that was used a lot in the earlier beginnings. I see that the flip-flop is the default, or one can select a different parameter. I believe I think it is the default now in the more recent colors. One of the things that I forgot to mention is that the latest, because there are, like I said, the automatic workflow from ONT, and I think the latest iteration of the best colors is called Bonito, and this adds 1.5% extra accuracy because they've refined even more the models. And bear in mind about that these models are trained in majority using bacterial DNA, and it turns out that it can differ the way that your input DNA, the source of your DNA, can impact the accuracy depending on the model that is trained upon a single type. Yes, it's the same DNA everywhere, but its source can have impacts. But yes, I hope I answered the question. Why use Medaka as well as Racon for long-read polishing? Well, I don't know if you imply that we should only use Racon and not Medaka, or only use Medaka and not use Racon. But I would say to use both, at least Racon, because it is specifically tailored to look at that miniasm output, and you should not use the raw miniasm output. It is a simple scaffold of long reads that really is a lot of errors. So you should at least use Racon on it, and Medaka, you should test it with your data. If it doesn't make sense, or if it's a lot of extra work, some people use it, some people don't use it. In our experience, we've used it because it's an extra tool in our kit to remove as many errors as possible. Does hybrid sequencing make a big difference to cost of sequencing? Yes, so it would, because you would then have to not necessarily double the cost, but you would incur costs for the same sample to have library prep and sequencing run in addition to your usual short read. So yeah, that would add cost for sure. Would you recommend a hybrid assembly, or does it depend on a downstream processing? If you have the short reads, it is the best way currently to produce the highest accuracies. So if you have them, definitely, and the processing requirements are not that high. BWA, even for large data, as well as Pylon, are quite scalable. And if you have the short reads, you should definitely use it. Do you know a training data set to practice at home with the tools that you teach us today? I could look into that. The purpose today was not to do that in real time with you, but I can definitely add and send an email to the attendees. There are a few. There is a ONT-led seminar and stuff like that, so I will duly credit them if I found, but there are packages and small data sets that definitely are out there, and I can definitely add them to the webinar resources page. Thank you for that suggestion. Oh, back to DNA and RNA sequencing. Do you think it will be possible to sequence both types of molecules simultaneously in the near future? I don't know. Maybe we should call ONT about that. I guess anything is possible, but right now, I think the software would have, you would have to calibrate the software for it to use the model and detect automatically what kind of molecule you're sequencing, because I don't think it would be good at differentiating the different types of molecules in real time. Interesting consideration. I know that they are working on even being able to sequence proteins directly. Considering that the average gene size of RefSeq phages are at about 600 base pairs or 200 amino acids, I was wondering how one was able to infer 98% accuracy when the average AA length was 100. Let me reread that. Right, so in the graph that I showed you with RACON, we did not use RefSeq. We sequenced with short reads our own virome sample, and we looked directly at the properties and the genomic features of that natural virus community, and we compared it directly to the same sample being sequenced with long reads, and this is where we saw these protein sizes. I hope that answered your question. Could you kindly share this pipeline and the data set to practice? Right, so I have put on the webinar resource page a link to all of these tools, most of which, if not all, are on GitHub. And we are working in the near future to try to make a something that is more convenient for people to use. But again, it is a bit challenging given the fast-paced nature of the updates and stuff like that. So there's a couple of considerations for a package that is polyvalent enough. Well, thank you. Appreciate it. I hope it was useful to you. Yeah, excellent question, and I should have emphasized that a bit more. There are other assemblers and there are other tools for QC which can be used for non-virus data. You can sequence eukaryotic DNA, microbial DNA, barcode these, and these would be the same tools to base call and quality filter. Now you will have, where it starts to differ quite a bit is in terms of assembly, where for microbial genomes, for example, a very powerful and well-documented assembler is Fly, which is sort of graph-based as well and has a metagenomic option, which they recently updated to be even more user-friendly in terms of the parameter requirements that they had in the past. But they also have a plasmid option, to look for these sort of smaller genome elements. We have compared Fly to the OLC method as well. And so I invite you to go and read the paper. I'm not against it at all. But again, I invite you to try it with your data first. And if you have short reads, to try to see what works best. And the computing power that you have as well. Fly is quite efficient. So yeah, but definitely the assembler choice matters most for the organism that you have, for sure. Can one use the pipeline to look for viruses in Illumina sequenced metagenome, not really sequenced, for metavirome? I see. So you're saying, yeah, so you could have a bulk metagenome, if that's what you mean, and correct all of these, create contigs, and apply a virus identifier on these corrected sequences. What you essentially do is try to at least get to a point where you have, puzzled the pieces together properly. And after that, you can totally use VirSorter or Vibrant or any other software like this that look for viruses in metagenome datasets. So definitely, yeah. Well, thank you. You're most welcome. All right. Thank you. Appreciate it. All right. Unless there are any more lingering questions, again, feel free to reach out to me in my email, on Twitter. Please have a look at the documentation we put on the Center of Microbiome Science website resources in the webinar section. And I think we can call it a day. What do you say, Kate? Oh, there's a question. Oh, that if there is a choice between Illumina short read versus Nanopore, which one would be best for microbial genomes assembly? It depends what you would like what is your research question and what kind of sample do you have. If you have an isolate in the lab, I would definitely go with Nanopore. If you have a metagenome, lots and lots of data and bins, I would go with short reads, definitely. And if you have some genomes that you feel are too fragmented or that you really want to hone in for your specific analysis, then I would take that sample and try to augment it with short reads. Long reads, I'm sorry.